Okay, it looks like we're live. So today I'll be reviewing this cell paper, the structure, function, and antigenicity of the SARS coronavirus 2 spike glycoprotein. And I will leave a link for this paper down in the description below. Excuse me. So let's start. So structure, function, and antigenicity of the SARS-CoV-2 spike glycoprotein. So as a quick reminder, the spikes of the coronaviruses are these things right here. They make the corona or the crown of the viruses. So here we have the original SARS coronavirus that caused an epidemic a few decades or quite a few years ago. And here we have the SARS coronavirus 2, which is going on, or which is the virus responsible for the current pandemic going on right now. And for both of these viruses, these spike proteins that are coming off of it both interact with the same receptor, and this is referred to as the ACE2 receptor. So these viruses bind to the ACE2 receptor, and this is the way that they stick to the cells, which then allows them or increases the likelihood of them being incorporated into the cell in the first place. So SARS-CoV-2 and SARS-CoV-3 bind with similar affinities to the ACE2 receptor. If you go into a little bit more detail in the paper, it's actually true that SARS-CoV-2 binds quite a bit more strongly, but similar meaning within the same order of magnitude, I would assume in this particular case. And structures of the SARS-CoV-2 spike glycoprotein in the two conformations. So there's going to be multiple conformations of the SARS coronavirus 2 spike protein, and this paper was able to show these two specific orientations based on their cryo-EM structure. And then in the end, the last highlight is SARS coronavirus 2 polyclonal antibodies inhibit SARS coronavirus 2 spike mediated entry into cells. And so here specifically polyclonal antibodies, meaning many different types of antibodies being used at the same time, can have a drastic effect on allowing the SARS-2 coronavirus from entering the cell. So these are the highlights of this particular paper. So without further ado, summary, and keep in mind this paper came out over a month ago, so it's a little bit outdated, but the emergence of the SARS coronavirus 2 has resulted in over 90,000 infections and 3,000 deaths, which, you know, a month later, those numbers are off by over an order of magnitude now. So coronavirus spike S glycoproteins promote entry into the cells and are the main target of antibodies. So glycoproteins specifically are proteins that have glyco, or they have small sugars that are added onto the protein itself. So that's why it's called a glycoprotein. And in this particular case, the glycoprotein is the spike glycoprotein, and we refer to this as S throughout the paper. So we should and are the main target of antibodies. So when we are producing antibodies for these viruses, very frequently it is these spike proteins that we are making antibodies against, because these are the most prominent ones, the most prominent proteins on the surfaces of the viruses. So continuing, we show that SARS-CoV-2 S spike uses ACE2, which is that receptor on your cells, to enter cells, and that the receptor binding domains of the SARS-CoV-2 S and SARS-CoV-S, which is the original SARS S protein, they bind with similar affinities to human ACE2. And similar is a relative term, so we are going to talk about that a little bit later. Correlating with the efficient spread of SARS-CoV-2 among humans. We found that SARS-CoV-2 glycoprotein harbors a furin cleavage site at the boundary between the S1 and the S2 subunits. So keep in mind that, I shouldn't say keep in mind, I've done previous videos about the furin cleavage site. So very frequently for viruses to enter cells properly, they need to have a cleavage site which then ends up breaking apart the protein, and that breaking apart often facilitates the virus being incorporated into the cell in the first place. So frequently, it doesn't need to be a furin cleavage site. It could just be any type of cleavage site, except the furin cleavage sites are usually a little bit more dangerous because your body produces furin throughout the entire body. So if you get infected with a virus that has the furin cleavage site, it might not just infect one particular type of cell or a few types of cells. Pretty much every cell that expresses the receptor for it to bind to, all of those cells become vulnerable. So for the SARS-CoV-2 S-glycoprotein, it has a furin cleavage site, and this was not present in the original SARS coronavirus spike protein. So just keep this in mind, and I did a previous video on furin if you want to go look at that video before continuing on with this. So, which is processed during biogenesis and sets this virus apart from SARS-CoV and SARS-related coronaviruses. So this virus that is causing the current pandemic is different in regards to this furin cleavage site with the original SARS and many other SARS that are closely related to it. Excuse me. So we determined cryo-EM structures of the SARS coronavirus 2 as ectodomain trimer. So ectodomain just means the part of the protein that sticks outside of the virus shell. So ecto meaning out, ectodomain, the domain that is outside. And it's a trimer, so these S proteins group in groups of 3. Providing a blueprint for the design of vaccines and inhibitors of viral entry. Finally, we demonstrate that SARS coronavirus S murine polyclonal antibodies potentially inhibit SARS coronavirus 2 spike-mediated entry into cells. So we demonstrate that murine is the genus of rodents, so rodent polyclonal antibodies potentially inhibit it, which would indicate success of antibodies and success of your body being able to produce antibodies against coronavirus in the long run. So, inhibits are indicating that cross-neutralizing antibodies targeting conserved S epitopes can be elicited upon vaccination. So here they're saying if we are able to make antibodies regarding these conserved S epitopes. So epitopes are part of proteins that an antibody is reactive against. And so what we're hoping is we can design antibodies that target the conserved parts of the S protein. And this will not just help us with this coronavirus, but these vaccines would be cross-neutralizing, which means they will help us with different coronaviruses as well that have these conserved S epitopes or these conserved regions. So without further ado, let's get into it. So 3 coronaviruses have crossed the species barrier to cause deadly pneumonia in humans since the beginning of the 21st century. These are severe acute respiratory syndrome coronavirus, or SARS-CoV, and Middle East respiratory syndrome coronavirus, MERS-CoV, and now we have the new SARS-CoV-2. So SARS-CoV emerged in the Guangdong province of China in 2002 and spread to 5 continents through air travel routes, infecting 8,098 people and causing 774 deaths. In 2012, MERS-CoV emerged in the Arabian Peninsula, where it remains a major public health concern even to this day, and was exported to 27 countries, infecting a total of 2,494 individuals and claiming 858 lives. A previous unknown coronavirus named SARS-CoV-2 was discovered in December 2019 in Wuhan, in the Hubei province of China, and was sequenced and isolated by January 2020. SARS-CoV-2 is associated with an ongoing outbreak of atypical pneumonia COVID 2019, and has affected over 90,000 people and killed more than 3,000 of those affected in over 60 countries as of March 3rd, 2012. So yeah, this is this is a very old paper and it's very out of date with its numbers. On January 30th, 2020, the World Health Organization declared the SARS-CoV-2 epidemic a public health emergency of international concern. So, MERS-CoV was suggested to originate from bats, but the reservoir host fueling spillover to humans is unequivocally dromedary camels. So, we assume that the original MERS virus is from bats, and it is harbored within bats, but it went from bats to camels first, and then now it's a disease that is spreading among camels, even to this day. But as long as we're careful, we know that the disease is in camels, so we know to avoid. We have learned ways to avoid getting the disease from the camels to this day. So both SARS coronavirus and SARS coronavirus 2 are closely related and originated in bats, who most likely serve as reservoir hosts of these two viruses. Whereas palm civets and raccoon dogs have been recognized as intermediate hosts for zoonotic transmission. So zoonotic transmission means that you can get it from animals, of SARS-CoV, the original SARS, between bats and humans. The SARS-CoV-2 intermediate host remains unknown to this day. So we still do not know. We still do not know if this came directly from bats to humans. We still do not know if it went from a bat into a different creature first, and then from that other creature to humans. Yeah, there's a lot of unknowns. I mean, it could have been a mosquito for all we know. Anyways, the recurrent spillovers of coronaviruses in humans, along with detection of numerous coronaviruses in bats, including many SARS-related coronaviruses, suggest that future zoonotic transmission events may continue. So this is not the first time that we've had a spillover event from bats, and most likely it will not be the last. So in addition to the highly pathogenic zoonotic pathogens, SARS-CoV, MERS-CoV, and SARS-CoV-2, all belonging to the beta coronavirus genus, 4 low pathogenicity coronaviruses are endemic in humans, and these are HCoV, OC43, HKU1, NL63, and this one also uses the ACE2 receptor to gain entry in the cell, just so you're aware, and 229E. To date, no therapeutics or vaccines are approved against any human-infecting coronaviruses. So this is unfortunate. I'll look into the literature to see if we have vaccines against coronaviruses that infect some of our livestock. We tend to be a little bit more, what would the word be, liberal with how much testing needs to be done before a vaccine is approved for animal usage. So it's harder to get a human vaccine through the FDA than it is to get an animal vaccine. And so some animal vaccines are a little bit more dangerous than human vaccines simply because it takes less trial to get them through. So coronavirus entry into host cells is mediated by transmembrane spike glycoproteins that form homotrimers. Homotrimers meaning it's a trimer of all the same proteins. So in contrast to a heterotrimer or a heteropolymer, then you would have different proteins that are coming together in one. But in this particular case, they are the homotrimers, so they're all the same. So S comprises 2 functional subunits responsible for binding to the host cell receptor. So the S1 subunit and fusion of the viral and cellular membranes, which are in the S2 subunit. So the S1 subunit and the S2 subunits are a part of the same spike protein, so spike sub 1 and spike sub 2. For many coronaviruses, S is cleaved at the boundary between the S1 and the S2 subunits, which remain non-covalently bound in the pre-fusion conformation. So even after these end up getting catalytically split apart, So you have your spike protein, and the spike protein has the S1 and the S2 subunits. Even after the protease splits these two apart, they still stick together in a particular way. They still stick together even after they've broken apart. And this process helps get the virus into the cell. So let's continue here. So the distal, or the farther away spike sub1 subunit, comprises the receptor binding domains and contributes to stabilization of the pre-fusion state of the membrane-anchored S2 subunit that contains the fusion machinery. So the S1 subunit will have what is known as the receptor binding domain. And this receptor binding domain is the domain or the part of the S1 subunit that allows the virus to stick to the receptor. And in this case, the receptor is the ACE2 receptor. So continuing, and contributes to stabilization of the pre-fusion state of the membrane anchored S2 subunit that contains the fusion machinery. So the S1 stabilizes the S2. That's all I can say. For all coronaviruses, the spike is further cleaved by host proteases at the so-called S2 prime site, located immediately upstream of the fusion peptide. This cleavage has been proposed to activate the protein for membrane fusion via extensive irreversible conformational changes. As a result, coronavirus entry into susceptible cells is a complex process that requires concerted action of receptor binding and proteolytic processing of the S protein to promote virus infection. And why would this be a strategy? So the strategy that the virus is employing is it wants to have one protein do multiple functions. And one can assume that the function of binding to the receptor is going to require one shape of your protein, but there might be a completely different shape of your proteins that will allow the that will allow the genetic material of the virus to enter the cell. So one of the best ways of accomplishing this is through proteolytic cleavage. So once you break the protein, you then create the opportunity to have a lot of different shapes. And one of the shapes helps the virus stick to the cell, and a different shape helps get the virus into the cell. So continuing, different coronaviruses use distinct domains with the S1 subunit to recognize a variety of attachment and entry receptors, depending on the viral species. So endemic human coronaviruses, OC43 and HKU, attach through their S domain A, so S sup A, to the 5N-acetyl-9-O-acetyl silicides found on glycoproteins and the glycolipids at the host cell interface to enable entry into susceptible cells. So the best way to describe this is I'll just do a Google search for y'all. So let's do Wikipedia. So this is the type of very complex sugar. And this complex sugar is on the surfaces of many glycoproteins and on the surface of many glycolipids on the cells. And so these two coronaviruses stick to these sialicides, or stick to these sugars, and that is one way for them to get close or stick to the cell and get into proximity. MERS coronavirus S, however, uses domain A to recognize non-acetylated sialicides, attachment receptors, which likely promote subsequent binding to domain B, or S sup B, to the entry receptor, dipeptidyl peptidase 4. So MERS employs a similar strategy. It sticks to these non-acylated sialicide attachment receptors, and then after it sticks, it then uses its location to then bind to these dipeptidyl peptidase 4 receptors. So in this particular case for MERS, it's actually using multiple receptors to get into the cell. So it's using multiple techniques to get into the cell. And so one could assume that this redundancy, this multiple abilities of getting stuck to a cell can facilitate in viruses or improve the fitness of a virus to be more pathogenic or be able to infect more cells. And if it can infect more cells, then it's going to produce more progeny. And so the viruses that are able to employ multiple techniques to get into a cell are going to be much more successful at duplicating and spreading further. So as the SARS-CoV and several SARS-related coronaviruses, SARS-CoV, interact directly with angiotensin converting enzyme 2. And so this, we love acronyms in biochemistry, so we just call it the ACE2 receptor, via their S-suit-B to enter target cells. So as the coronavirus spike glycoprotein is surface exposed and mediates entry into host cells, it is the main target of neutralizing antibodies, or ABSs, upon infection and the focus of therapeutic and vaccine design. So when we are developing therapeutics and we are developing these pharmaceuticals that can help people avoid getting more and more sick. A lot of times, especially when the virus is in its replicating phase, we employ therapeutics or pharmaceuticals that stop or that interact with this spike glycoprotein. And the reason why is because since it's exposed on the virus, it's an easy target for drug design. And likewise for vaccine design, because when we design vaccines, we would like the vaccines to create antibodies, and an easy target for those antibodies are the shapes that we see on the S protein. So S trimers, or spike trimers, are extensively decorated with N-linked glycans that are important for proper folding. So N-linked glycans, so glycans are small sugars or oligosaccharides, and N-linked means that these sugars are connected to nitrogens, and proteins have a lot of nitrogens that are sticking off of them. And so these are prime places to stick glycans or stick small sugars or small oligosaccharides onto. And these glycans, or this glycosylation is the process. This is important for proper folding. It's also good at protecting certain areas. It's also important in telling where the cell should be putting proteins. So it's a way to communicate with the environment what should happen to the proteins that end up getting glycosylated and then get turned into glycoproteins. So N-linked glycans that are important for proper folding and for modulating accessibility to host proteases and neutralizing antibodies. So another problem, I shouldn't say a problem, but another technique of these viruses and of many pathogens is they utilize their host. They utilize the enzymes of the host to glycosylate their proteins. And the problem for you as an individual is that it's more difficult for your immune system to recognize a protein if your own body has made the sugars sticking on that protein. So I like to think of this as think of people with ABO blood types. So you can have A blood type, you can have B blood type, and you can have AB blood type. And I don't remember if it's N-linked or O-linked, but just keep in mind, if you are given the wrong type during a blood transfusion, your body can start attacking the red blood cells of the transfusion and can cause clotting throughout your entire body. And this is because your own body produces its own glycoproteins. And your own body recognizes its own glycoproteins. It recognizes the sugars that is put onto these proteins. So with the ABO blood type, if you have A blood, then your body and your cells are putting the A sugars on your proteins, or on the proteins of the red blood cells. So if you're given the blood type A in a blood transfusion, your immune system sees these A sugars and recognizes them as very similar to your own A sugars. And so it doesn't elicit the same immune response. However, if you're given a B antigen, or given blood from someone with type B blood, that type B blood is expressing a completely different sugar. And so it's very easy for your immune system, or the immune system of the A type, to see this blood sugar and say, this is not right, this is not me, and then elicits a response against it. So that's just one example. And the reason why an A type can accept blood O type is because the O red blood cells don't have any A's or any B's. So it's harder for your immune system to recognize those red blood cells as foreign, because they're not exposing any of those sugars. So this is one of, if not many of the techniques that viruses use for these N-linked glycosylation events, is that it modulates the accessibility to host proteases and neutralizing antibodies. So I'll also talk a little bit how these glycosylation events can even protect certain areas of the protein from being destroyed or cleaved. So we previously characterized potent human neutralizing antibodies from rare memory B cells of individuals infected with SARS-CoV or MERS-CoV in complex with SARS-CoV spike and MERS-CoV spike to provide molecular level information of the mechanism of competitive inhibition of S-B attachment to the host receptor. So they're just saying in a previous experiment, they were able to prove that these antibodies are effective, and the S230 anti-SARS-CoV antibody also acted by functionally mimicking receptor attachment and promoting S-fusogenic conformational rearrangements through a ratcheting mechanism that elucidated the unique nature of the coronavirus membrane fusion activation. So stepping back, this basically means that this S230 anti-SARS coronavirus antibody from the original, so it's an antibody that was functioning against the original SARS outbreak. This antibody mimicked, or I'll just say it mimicked the original ACE2 receptor to a degree. So then this antibody, which is not connected to a cell, ends up interacting with the virus, and it confuses the virus and makes the virus think, I'm already connected to a cell. And so then the protein undergoes its conformational changes. So the protein changes shape, thinking it's time to infect. But since the antibody is not connected to a cell, it doesn't infect and nothing happens. So this is one strategy that antibodies employ, or that your immune system employs to neutralize the virus. Anyways, we report here that ACE2 could mediate SARS coronavirus 2 spike-mediated entry into cells, establishing it as a functional receptor for this newly emerged coronavirus. The SARS coronavirus 2 S-B engages human ACE2 and frequently, so a lot of these ACE2 proteins are not just in humans, they are found in a lot of mammals, and in fact, I think ACEs are found throughout insects as well. So whenever we specify that this is the ACE2 receptor within humans, we denote it with this lowercase h. So ACE2 is just the human ACE2. And oftentimes we can find these receptors in completely different animals, but they might not necessarily be called ACE2. They might be called a metalloprotease or random. They might not necessarily have this exact same name. So the SARS coronavirus 2 SB engages human ACE2 with comparable affinity to SARS-CoV S-B from viral isolates associated with the 2002 and 2003 epidemic. So binding with high affinity to human ACE2. So here they say it's comparable affinity, and we'll see that that's not exactly the case, but comparable is a relative term, so they can definitely use that in this paper. So tight binding to human ACE2 could partially explain the efficient transmission of SARS coronavirus 2 in humans, as well as was the case for SARS coronavirus. We identified the presence of an unexpected furin cleavage site at the spike sub 1, spike sub 2 boundary of the SARS coronavirus spike, subunit 1 and subunit 2. So, as I said earlier, proteolytic cleavage or breaking apart proteins is sometimes extremely important for allowing the virus to get into the cell. And I did another video on furin cleavage sites. So in the original SARS spike protein, it did not have this furin cleavage site. It had what was known as a monobasic or dibasic, I don't remember which one, but it was just a basic cleavage site. And this basic cleavage site is very specific. It requires very specific proteases to attack it. But here in the SARS coronavirus 2, we have a furin cleavage site instead of that monobasic cleavage site. And this is why it's called unexpected, because this isn't in any of the closely related coronaviruses to SARS-CoV-2, which is cleaved during biosynthesis. So a novel feature setting this virus apart from SARS-CoV and SARS-related coronaviruses. So so with the original SARS coronavirus, it had a monobasic cleavage site, and this during the production of the original SARS virus. Very frequently, this site was not already broken apart. However, with the research of this particular paper, they say that as these viruses are being made in your cells, a lot of these spike proteins are already cleaved by furin. So they're pre-broken apart within the cell. Now, this could be positive or negative, but in this particular case, it seems to be a positive thing for this new SARS coronavirus, so SARS-CoV-2. So, as I said, this is a novel or a new feature setting this virus apart from the SARS-CoV and the related coronaviruses. Abrogation of this cleavage motif moderately affects SARS-CoV-2 spike-mediated entry into Vero E6 or BHK cells, but may contribute to expand the tropism of this virus, as reported for several highly pathogenic avian influenza viruses and pathogenic Newcastle disease virus. So the Vero E6 and the BHK cells, these are just, so when we are working in a lab, we don't often work with animals. Sometimes we just use what are referred to as cell lines. So we will have cells that we have taken away from a model organism, and then we can culture these cells. And then after we culture these cells, it makes it easier for us to have reproducible experiments being done on these cells versus having them be done on animals or done on insects or done on plants. So Vero E6 and BHK are just two different cell lines. And I should also mention that if you have cells that you're culturing, it is easier to do genetic modifications on those cells. So you can do a genetic modification on that cell, and then that can tell you whether or not the drug that you want to test or that the protein you want to test, if there's a difference depending on which genetic engineering you did to these cells. So, and these are experiments that are very difficult to do with full animals because then you have to raise an animal and you need to control a lot of the variables for those animals. And at some point, it becomes very costly to do a lot of these tests. So, in this case, the Vero E6 is a is from monkeys, if I remember correctly. Yeah, so Vero E6, they are, the Vero lineage is isolated from kidney epithelial cells from African green monkey. So these, since they're from monkeys, they should have a lot of the similar enzymes as humans. So this is to emulate or help recreate a human cell line as well. And for the BHK cells, this is going to be a little bit sad, but these are baby hamster kidney cells. So yeah, just keep that in mind. BHK is baby hamster kidney cells, and so we probably killed a baby hamster and cultured the cells from their kidneys, and those cells are still used today for experiments. But may contribute to expand the tropism of the virus. So this particular cleavage motif, sometimes it can make viruses more infectious, or maybe it's possible it could make a virus less infectious or less able to enter a cell. So if there's a lot of cleavage that is happening within the cell before it's produced, maybe it's not as effective at infecting a new cell. However, one of the benefits for the virus, not you, but one of the benefits for the virus is that if it has this polybasic cleavage site, it can then infect not just specific cells, but it can infect many types of cells because the enzyme that is used to break apart this fusion or this cleavage site is found throughout the entire body. The term would be it's ubiquitous. It's ubiquitous throughout the entire body. So when they say it can affect the tropism of the virus, what they're saying is it's affecting which cells or which tissues are being infected. And this is for a lot of highly pathogenic viruses. A lot of times they take advantage of this polybasic or furin cleavage site. And the reason why is if you can infect all of the cells of a creature, then it is easier for you to not just make more virus, to duplicate more. But then it means that you can spread to a lot of other places, which means there are more opportunities for the victim of the virus to experience pathogenicity or to experience illness, because it's not just in one small tissue, it's happening throughout the entire body. So, and that is what we're seeing with the current SARS-2 coronavirus going around. So, we determined cryo-electron microscopy or cryo-EM structures of the SARS-CoV-2 ectodomain trimer and revealed that it adopts multiple S-B conformations that are reminiscent of previous reports of both the original SARS spike and the MERS spike. Finally, we showed that SARS-CoV-S mouse polyclonal sera potentially inhibits entry into target cells of the SARS-CoV-2-S pseudotyped viruses. So this is a lot. What they're saying here is that the antibodies for the original SARS-CoV-S, if you have polyclonal antibodies, if you have a lot of different types, so polyclonal, many clones, if you have a lot of different antibodies for the original SARS-CoV-S, and if you can possibly have what would the word be? Immunity. You could possibly have immunity from this new SARS coronavirus S. And the reason why is there are some locations on the original SARS spike protein that are similar to this new SARS-CoV-2 spike protein. So, and they say pseudotyped viruses because they didn't actually infect, they didn't actually use this highly pathogenic SARS-CoV-2. What they did was they took a pseudotyped virus or a weakened virus that is completely unrelated to SARS-CoV-2. And then they put in the spike protein of SARS-CoV-2 and tested to see how effective it was at infecting. So this is one way that this particular lab is able to not have to handle this new SARS coronavirus. They can just do the research on a highly weakened form of a completely different virus and then give it just this one part of the new virus to see how it works. So let's go to the results. And I'm going to go over these figures later, but I just want to go through a lot of the text first before I go over them. So the results. ACE2 entry, or ACE2 is an entry receptor for SARS coronavirus 2. So the SARS-CoV-2 S glycoprotein shares 76% amino acid sequence identity with SARS-CoV S urbani and 80% identity with bat SARS related coronavirus ZXC21S and ZC45S glycoprotein. So that doesn't mean much. Just know that it shares a lot of sequence identity with these two other SARS coronaviruses. Anyway, the latter two SARS-related coronavirus sequences were identified from Rhinophis sinicus, or this is the Chinese horseshoe bats, the species from which SARS-CoV these were isolated. Furthermore, and just so you know, that's the pretty ugly looking bat that sort of looks evil, but I guess beauty is relative. So furthermore, recently reported that, this person, Zhao, recently reported that SARS-CoV-2 is most closely related with the bat SARS-related coronavirus RATG13, and with which it forms a distinct lineage from other SARS-related coronaviruses, and that their S glycoprotein shares 97% amino acid sequence identity. SARS coronavirus. So they're basically saying between this bat coronavirus and the current coronavirus that is going around right now, the spike protein in of itself shares about 97% amino acid sequence identity. So it's about 97% the same. So SARS-CoV-2 recognizes its entry receptor, human ACE2, at the surface of type 1 pneumocytes. Pneumocytes, I believe, are lung cells. Yeah, so the surface epithelial cells of the alveoli, which are the little sacs within your lungs. So, which shares 75, which shares 75% overall amino acid sequence identity with SARS-CoV-2, S-SUP-B, and 50% identity with their receptor binding motifs. And so, lost my train of thought a little bit here, but SARS-CoV-2 recognizes its entry receptor at the human. So they're saying with the original SARS, it also binds with the human ACE2 receptor of these type 2 lung cells or alveoli cells. And it shares about 75% overall amino acid sequence identity with this new SARS that is, or this new SARS-CoV-2, and 50% identity with the receptor binding motif. So this receptor binding motif is a small part on the spike protein that motif is the part that's doing the work of binding with the ACE2 receptor. So here they're saying for the entire spike protein, The old SARS and the new SARS have about 75% sequence identity. However, in the one little part of that spike protein, that one part that binds between the spike protein and the ACE2 receptor of the victim cell, that only has 50% sequence identity. So one could assume, okay, there's quite a bit more change when it comes to how they're interacting. Continuing, previous studies also showed that host proteases cathepsin L and TMPRSS2 prime, TMPRSS2, these two proteases prime the SARS coronavirus spike, so the original SARS, for membrane fusion through cleavage at this S sub 1, S sub 2 junction and at the S sub 2 prime sites. So here, as I said earlier, you need these specific proteases, TMPRSS2 and cathepsin L. These are the two proteases that are known to cleave the spike protein of the original SARS. So it cleaves it, excuse me, and then after that cleavage, the cell's allowed to get into, or the virus is allowed to get into the cell. But we will learn in the case of this new coronavirus, these two specific proteases that are found in the lung tissue and in only a few cells, they are not necessary for breaking apart the new SARS-CoV-2 spike protein. So just keep that in mind. So we set out to investigate the functional determinants of S-mediated entry into target cells using a murine or a rodent leukemia virus or MLV pseudotyping system. So this is just a weakened, this MLV is a weakened virus that is missing a lot of really important replication. It's just a very weak virus, and a lot of the parts are missing. And so sometimes we can add parts from different viruses to replace some of the parts that have been taken away from this MLV virus. And one of the benefits of having a model virus is that it's reproducible. So you can use the same system in a lot of different experiments in a lot of different labs. And it's one way to be safe when doing virus studies, because it's dangerous to be handling viruses in the lab. So this is a way to have a safe system, especially one that might not be very good at infecting humans. If it's only infecting murine, which murine means rodent, if it only affects rodents, then at least that's a little bit safer than dealing with a human virus. Anyways, to assess the ability of the SARS-CoV-2 spike to promote entry into target cells, we first compared transduction of SARS-CoV-2 spike MLV and SARS-CoV SMLV into the Vero E6 cells. So remember, this is the model cells from the monkey, and these cells are supposed to, or should have, very similar enzymes and very similar proteins as a normal human cell, because it's from a monkey. So they're supposed to be very similar. And they compare this weakened virus with the SARS-CoV-2 spike protein, and they're comparing it to the weakened virus that has the original SARS spike protein. And the reason why we are using this Vero E6 cells is that they are known to express ACE2, and we know already that they support SARS-CoV replication. So we already know that this cell line from monkeys can duplicate or are susceptible to SARS, or the original SARS. But we're not testing it with the original SARS. We're testing it with these SARS-CoV spike MLV viruses or weakened viruses instead. So it's functioning as a control. So both pseudoviruses entered cells equally well, suggesting that SARS-CoV-2 MLV could use African green monkey ACE2 receptors. So remember, these are African green monkeys are the source of these Vero E6 cells. We are presuming that it uses the ACE2 as the entry receptor. To confirm these results, we evaluated entry into BHK cells and observed the transient transfection with human ACE2 rendered them susceptible to transduction with SARS-CoV-2 spike MLV. So these BHK cells, which come from the baby hamster kidney cells, these ones, I don't know if it's normally the case, but they do not express the ACE2 receptor, or if they do, they have done genetic engineering to remove their ACE2 receptors. And since we know that these original BHK cells do not have the receptor, we can test to see if our virus that we've produced is able to infect them. And it's not able to infect them normally. However, if we genetically engineer them to express the human ACE2 receptor, then they do end up becoming susceptible to our new virus. So what that confirms for us is that the new SARS coronavirus 2 spike protein, even on a weakened virus, is able to infect a previously immune cell. It's able to infect it if we give them the ACE2 receptor. So this verifies that the ACE2 receptor is a receptor that the SARS coronavirus 2, or the new coronavirus, is able to exploit to get into cells. Now, that doesn't say that that is the only receptor. There could be another receptor that any virus could use. But this is at least one receptor that will allow the virus to stick to the cells. 52 minutes. So continuing, these results demonstrate that human ACE2 is a functional receptor for SARS coronavirus 2 in agreement with recently reported findings. So here, I'm going to go up to the graphs, or to the data where they show this. So here is a graph of the Vero E6 cells, and here's a graph of the baby hamster kidney cells. So this is just a graph of the amount of the activity that they have seen, or how many. I think RLU means relative units, but just ignore it. Just know that this is the amount of viruses that end up getting in. And here they say it's the pseudovirus entry. So how many pseudoviruses are able to get into the cell? And when you just have the cells only, you should have nothing getting in. Okay? Now, if you have these pseudoviruses genetically engineered to express the SARS coronavirus 2 spike glycoprotein, then it is able to get into these monkey cells, the Vero E6 cells. It's able to get into them. And as a control, they didn't just use the new spike protein of the new coronavirus, they used the spike protein of the old coronavirus as well for the original SARS. And that also allowed our pseudoviruses, to get into these monkey cells. And this is for later. And to verify whether or not it was using the ACE2 receptor, we didn't use the monkey cells. We used these baby hamster kidney cells, and they might have been genetically engineered to not express ACE2 at all. Regardless, the cells alone, nothing. When we give them the virus with the SARS-CoV-2 spike protein, nothing gets in. So what we do is we genetically engineer these BHK cells to have the human ACE2 coronavirus on them. And lo and behold, when you do that, you end up creating the conditions which let your pseudovirus to enter. So this tells us that ACE2, human ACE2, is the, if not one, just one of the receptors that the new SARS coronavirus can use. So, going down. So, sequence analysis of SARS-CoV-2 spike revealed the presence of 4 amino acid residue insertions at the boundary between the S sub 1 and the S sub 2 subunits compared with SARS-CoV, the original SARS-CoV spike, and related SARS-CoV spikes. This results in the introduction of a furin cleavage site, a feature conserved among the 144 SARS-CoV-2 isolates sequenced to date, but not in closely related RATG13 spikes. So what they're saying is this furin cleavage site is a feature that you can find in a lot of different coronaviruses, up to 144 SARS. So what they're saying is this furin cleavage site is a part of and is conserved in all of the SARS-2 coronaviruses that we've sequenced. So you might have seen that there are many different strains of this new coronavirus that are going around. In all of the ones that we've sequenced as of yet, this 4 amino acid sequence has been conserved. So you can find it in every single SARS-CoV-2 that we've sequenced as of yet. They have this furin polybasic cleavage site. But this site is not in the closely related spike proteins of the bat coronaviruses. So using Western blot analysis, we observe that the SARS-CoV-2 spike was virtually entirely processed at the S1, S2 site during biosynthesis in this particular cell line. And this is presumably by furin, which is the protease. It's the enzyme that finds this sequence and breaks it apart. Presumably by furin in the Golgi compartment. This observation contrasts with the original SARS coronavirus, which was incorporated into the pseudovariants largely uncleaved. So a lot to unpack there, but let's go to the figure. So given the pseudoviruses that we make, and let's say we culture them in a cell and we want to do an experiment to see how are the proteins being processed after they've been made. So what post-translational modifications have been made? And I've done a previous video on that as well. So when we have the original SARS and I should also mention, so this is a page gel, or it's the Western blot, I should say. So what they did was they took the viruses and then they split apart, or they separated all the proteins from each other. And then they ran these proteins on a gel. And what this gel does, they apply a voltage to it, and then the voltage is pushing the proteins throughout the gel. But different proteins are different shapes. And so they will separate based on shape. And you can see that over here, where kilodaltons is just a, I shouldn't say shape, I should say size. So it's separating them based on size, and possibly shape as well. And here, kilodaltons, this is just a measurement of how large, or the mass of the proteins are. So with the original SARS coronavirus S protein, as they're being synthesized, as the viruses are being synthesized, and if you break apart these viruses and you look at the proteins, all of these proteins are large. They are between 150 and 250 kilodaltons. So this is the spike protein in its entirety, if it's full. So, and this is the original SARS coronavirus. They did it as well with SARS-CoV-2 spike protein, and when they ran those proteins through this gel, normally, if it was uncleaved, it'd be here. But most of them are down here, meaning that they have been split apart. So rather than having this large 150 to 250 kilodalton pieces, we end up having a smaller piece, which means that a large part of the spike protein has been removed in some way. And so when they collect the viruses and they run the analysis, rather than having a bunch of spike protein up here, they end up having a bunch of smaller parts of the spike protein. Now, that doesn't mean it is possible that you still have some of the uncleaved spike proteins somewhere in here. It's just that there's a lot less of them and you can hardly see them. And I should also mention that even if it's only part of the spike protein, the part that sticks to the cells is still on the spike protein. So I should say the binding domain of the spike protein is still present on these proteins even after the cleavage event. So just keep that in mind. So, and then continuing, when they did a mutation on the SARS coronavirus 2 pseudoviruses, they saw that most of the spike, when incorporated into the new virus, most of those spikes were still full. Most of them were still complete. So, going back down. Oh, so basically what this would indicate is that the furin, we don't know if this is physiologically what's happening in people, but it is presumed that these spike proteins are being cleaved even before the virus leaves the cell after being produced. So what they're saying is there might be a lot of viruses that if someone coughs them out, a lot of those viruses are going to be pre-cleaved. They're already going to have a part of their spike protein missing. Now, remember, these are in trimers, so maybe some of the spike proteins, they form trimers, right? Maybe some of them are missing, and maybe only one of them is still sticking up or still full. And remember, these spike proteins, they're all over a virus, so it's very likely that a virus with their hundreds of spikes on them might still have quite a few uncleaved spikes on top of them. So just keep that in mind. You know, it's all a messy process. It's all, these viruses form, they're just grabbing the closest thing to them that works, right? So as they form, they could be grabbing some uncleaved and cleaved. Yeah, so let's continue. This observation contrasts with SARS-CoV-S, which was incorporated into the pseudovirus to study the influence on pseudovirus entry of the SARS coronavirus 2 S1, S2 furin cleavage site. We designed an S mutant that's lacking the 4 amino acid residue insertion and the furin cleavage site by mutating this particular amino acid sequence. So here, this this entire area has been replaced with these amino acids. So each of these letters are a different amino acid. So R's stand for arginine. So arginine, arginine, alanine, arginine. This is the stereotypical polybasic cleavage site of furin. This can be cleaved by furin, and the cleavage happens here where this arrow is at. So to see, they want to see if by removing this, if the virus particles that result from this, they want to see if the spikes were whole long spikes in the viruses that are being produced. And here, as seen by the figure here, it works. So they remove that site, and then the spike protein exists in its full large form. So, going down. And so here you can see they remove these 1, 2, 3, 4, 5, 6. They remove these 6 amino acids, the N through the A, and then they replace them with these 2 amino acids. And so now it's not a polybasic cleavage site, it's just a monobasic cleavage site. So, going back, SARS-CoV-2 S furin mutant preserved only the conserved arginine residue at position 685 of the wild-type SARS coronavirus 2 S, thereby mimicking the S sub 1, S sub 2 cleavage site of the related SARS-related coronavirus S spike CZX21. So they are mimicking the cleavage site of a closely related coronavirus. This is therefore expected to undergo processing at the S1, S2 site upon encountering a target cell, similar to SARS-CoV-S and this related coronavirus, through the TMPR SS2 and/or through cathepsin L. So they removed this polybasic cleavage site and they replaced it with the monobasic cleavage site. But they presume that this monobasic cleavage site is still susceptible to being cleaved. It just can only be cleaved by these specific proteases, or at least preferentially by these specific proteases. So as expected, SARS coronavirus 2, MLV, so the virus with this new spike protein, it harbored the uncleaved S protein upon budding, or upon the formation of these new viruses. They had the full spike protein, uncleaved. And then the observed transduction efficiency of the Vero E6 cells was higher for the SARS-CoV-2 S furin, or these mutated cells, than for the SARS-CoV-2 spike MLV, the normal ones, without the genetic engineering at this furin site. So we can see in the monkey cells, which express the ACE2 receptor and also express furin, and then also express these specific proteins that can do cleavage. They took these viruses that had the furin mutant on them, and they actually got an increase relatively of how many were able to get in. And then they took those same viruses, and then infected the baby hamster kidney cells, and they also were able to go in, except with less efficiency. So what they're saying is, and it's hard to, even I shouldn't say even I, but I have a hard time grasping all of this. It's saying that the unbound or the uncleaved spikes, even though they are cleaved normally beforehand, even the uncleaved ones are able to enter the cell, or able to get into the cell without any problem. And if you remove the polybasic cleavage site and replace it with a more specific cleavage site, it is still able, with a different protease, to get into the cell, and sometimes with very good efficiency. In this case, it was also able to do that with the BHK cells that are expressing the human ACE2 receptor, granted with a little bit less less ability to do so. Now, what this could tell you is that these very specific cells that we know express the proteases that work on the monobasic cleavage sites, these, since they express a lot of it, are fine, and since it's closely related, they're close enough to work well with this genetically engineered virus without the furin cleavage site. But in these baby hamster kidney cells, maybe they don't have the very specific monobasic cleavage site that the monkeys do, so maybe there's a species barrier there, they weren't as able to cleave this virus. However, when they had the original, when it had the polybasic furin cleavage site, it then brought, um, it created the ability for this virus to infect a larger range, or easily infect a larger range of species. So this is a strategy that the polybasic cleavage site can afford viruses, is that it allows viruses to be less precise in what species they're trying to target. So, and here you can see, you get rid of the polybasic cleavage site, it can actually improve the ability for this virus to infect a monkey cell. But it decreases the ability to infect, wait, I gotta make sure that I said that correctly. So, even if you remove the furin, the virus is still able to infect this specific cell. And then if you remove the furin, it is less able to infect a more distantly related species of the original host, which in this case is a human. But if you have the furin cleavage site, well, they're a lot more similar. Now, granted, I'm spitballing right now, and these are just they're relatively closely related species already. We're not too different from hamsters, but this is one example that helps support the hypothesis that the furin cleavage site does end up conferring a lot of general pathogenicity to a wider range of species. So, going back down, and we're at, an hour 11. Damn. So, SARS-CoV-2, wait. Are we already? Oh, wow, we're already down there. So, cleavage is not. So these results suggest that the S1, S2 cleavage during spike biosynthesis was not necessary for spike-mediated entry in the conditions of our experiments. So they were clear to say that in the conditions of their experiments, this is happening. That's not necessarily what is happening in the real world. So we speculate that the detection of a polybasic cleavage site in the fusion glycoprotein of the SARS coronavirus 2 could putatively expand its tropism and/or enhance its transmissibility compared with SARS coronavirus and SARS-related coronavirus isolates due to the near ubiquitous distribution of furin-like proteases and their reported effects on other viruses. So, just because furin and furin-like proteases, are expressed throughout the body and expressed in many species, we say that this is going to help the virus become much more transmissible. So continuing on, SARS coronavirus 2 recognizes human ACE2 with comparable affinity to the SARS coronavirus. So the binding affinity is basically, you want to quantify the ability of, or how strong the bond is between two molecules that they're not covalently bonded, they're not forced to stick together, but the binding affinity means how much affinity is between them. So all the molecules in the universe are all jiggling around with thermal energy. Now, as they're jiggling around, sometimes they'll jiggle and they'll stick together. And then if they have a high affinity, they will stick together for quite some time before the random thermal energy ends up knocking them back away. So here they're doing an experiment to see if you take the ACE2 receptor, the human ACE2 receptor specifically, and you take the spikes of the coronaviruses, what is the binding efficiency or the binding affinity of the human ACE2 receptor and these proteins? So, continuing on, Oh, yeah. The binding affinity of SARS coronavirus for human ACE2 correlates with the overall rate of viral replication in distinct species, as well as with transmissibility and disease severity. Indeed, specific S-B mutations, which is where the binding domain was at, enabled efficient binding to the human ACE2 of SARS coronavirus isolates from the three phases of the 2002 to 2003 epidemic, which were associated with marked disease severity. So, what they're saying here is that if you control for all other variables and you have the spike protein of the original SARS epidemic, there were some mutations that happened on the spike protein. And they did affinity tests on those. And what they found is the stronger the affinity, the more that the spike protein stuck with the ACE2 receptor, if that strength was associated with more or a more pathogenic version of the disease. So this this might not be the case with all viruses, but for the case for the original SARS outbreak, the binding affinity was associated with how serious the disease was for the individual who got that particular strain of the virus. So in contrast, SARS coronavirus, the original SARS isolates detected during the brief 2003-2004 reemergence interacted more weakly with the human ACE2 receptor and had low pathogenicity and transmissibility. So what they're saying is when the spike actually very loosely interacted with the ACE2 receptor, then you could see the results of that weak interaction as a low pathogenicity or less symptoms in the second outbreak that happened. So one could argue that the SARS evolved to be less pathogenic, and that might have contributed to it might have been more asymptomatic, and so it might have been able to spread more easily, which is why you had a reemergence of the original SARS the next year. So this could, politically wise, I mean, I don't want to get into politics, but this means that as long as you are providing selective pressure against these coronaviruses, the selective pressure could, in fact, help evolve viruses to become less pathogenic. Because the less pathogenic and the more asymptomatic they are, that means that how do I phrase that? That they're going to slip through your evolutionary pressure to get rid of all the highly disease-ridden people. Or if someone shows symptoms, it is easier for you to catch that person, and then it is easier for you to prevent that person from spreading the disease, right? So that would be a selective pressure that society can put on a virus, which is why it is so important that us as a society, that we provide this selective pressure on this current pandemic that is going on. Because if we let it just roam through society without stopping infected people or stopping symptomatic people from spreading the virus, then the virus could easily mutate to bind stronger and stronger to the ACE2 receptor, or it could easily mutate into more and more pathogenic, more symptomatic strains. But as long as we're putting the selective pressure on the virus at all times, we are in fact selecting for more asymptomatic variants. We are selecting for weaker forms of the coronavirus. Now, granted, this is just for the acute or the immediate effects of the virus. Perhaps maybe there are some long-term consequences of being infected by an asymptomatic version of the coronavirus. So for instance, multiple studies have shown that half of asymptomatic people who contract this coronavirus still have the ground glass opacities or still have the signs of pneumonia in their lungs. And we still don't know what the long-term consequences of that are. We also don't know what the long-term consequences of the antibodies that we produce. We don't know those long-term consequences. So what I'm saying right now is it's good that we have selective pressure against having this virus spread like wildfire through our communities. But this is only selecting for the acute symptoms, but it's not selecting against the long-term symptomology. So I don't want to give complete or false hope that, oh yeah, we'll just all get it, but we'll all get the most benign version. That might not necessarily be something that you want still. So continuing, to understand the contribution of receptor interaction to the infectivity of SARS coronavirus 2, we characterized engagement of the human ACE2 by SARS-CoV-2 SV or S-SUP and SARS-CoV S-SUP side by side. So we used biolayer interferometry to study binding kinetics and affinity of the purified human ACE2 ectodomain. The ectodomain is the part of the human ACE2 receptor that sticks outside of your cells. So they just took the outside portion to do this experiment. To the SARS coronavirus 2 SUP, and these are, I'll just say the binding domains, and the SARS coronavirus original binding spike domain immobilized at the surface of biosensors. So this will be hard to understand unless you see a visual, so I'm going to do the Google search of this. So my favorite, let's go to the images. So this is my favorite image to show it. Load, please. Now, you know what? I'll just zoom in. No. So here, this is your sensor, and this is the membrane. What we do is we first connect one of the molecules that we want to test. So remember, we want to know the affinity. So we'll have one of the molecules that are connected to the membrane, and then we are going to add a solution with the other part afterwards. So here we load it, and in this case we are going to load our machine with the with the spike protein binding domains. So we'll do one of these for the new coronavirus, and we're going to do one of these for the old coronavirus. And so when you do this, you end up changing the baseline or changing the signal that you get. But after a while, all of your parts, all of your locations are full of your spike protein, and then you'll have your base signal. Then what we do later is we will add the other portion in different concentrations. And when we add it, when we do a high concentration, we'll see that there is a high signal, and eventually as many parts get full as possible. And if you do a lower concentration, then it'll not fill as many parts. And if you do a lower concentration, it'll be even less. So we do this until it's about flat. And this can tell us, relative signals between concentration. And if we decide to change what red portion we have here, you might have completely different numbers given the new concentrations of the other part. So this is the association part. But then about halfway through, we do the dissociation part, which is when we remove we wash it. We get rid of all of these. In this case, it's going to be the ACE2 receptors. We remove all of these, and then we watch as the ones that were bound originally to our membrane, eventually they're going to come off slowly. And then you can see the signal go down over time as they're being removed. So this is the general principle of the experiment. So here, they used biolayer interferometry to study binding kinetics and affinity of the purified human ACE2 ectodomain and the SARS coronavirus 2 spike binding and the original spike binding immobilized at the surface of the biosensors. We found that human ACE2 bound to SARS-CoV-2 spike soup binding and SARS-CoV spike subbinding with respective equilibrium dissociation constants of 1.2 nanomolar and 5.0 nanomolar. So best way to describe this is the equilibrium dissociation constant. If I'm remembering this correctly, it might not be the exact same definition, but given a certain number of your spike proteins, what concentration, so remember they're all jiggling around, what concentration of the ACE2 receptor do you need in the solution to have about half of your spike proteins to be covered? So nanomolar, just know that that is a measurement of concentration. When it comes to the SARS coronavirus 2 spike binding domain, it needs 1.2 nanomolars of the ACE2 receptors floating around for half of your spikes to be covered. However, when it comes to the original SARS coronavirus 2 spike domain, that requires 5 nanomolars of ACE2 receptors floating around for half of them to be occupied. So what they're saying is, you need about 4 times as much ACE2 receptor to bind with half of the original spike glycoprotein of the original SARS, and you only need 1.2 of that to have the same effect on this new coronavirus. So that's telling you that the new coronavirus spike protein is significantly better at binding the ACE2 receptor. Now here they say that they're comparable, but comparable, you can compare anything, right? So they're comparable in that they're within the same order of magnitude, I guess, but I guess they're trying not to be too dramatic in their paper. Continuing, and comparable kinetic rate constants. So kinetic rate constants means the speed at which you reach equilibrium. So apparently the rate at which the receptors are binding, those rates are about the same at the beginning, but we will learn, I'll show you in the graphs later, we'll learn that the the SARS-CoV-2 is actually significantly better and can create a much higher signal than the SARS-CoV-1. And although the off rate were slightly higher for the original SARS spike protein, the affinity determined here for SARS-CoV spike binding to human ACE2 is in line with prior studies, and the tighter apparent interactions formed by SARS-CoV S soup binding compared with the SARS-CoV S sub1 subunits or the SARS-CoV spike trimer. So what they're saying is, when you are just dealing with this binding domain, it interacts a little bit better than if you have the entire structure. But they're trying to, as long as everything's being controlled and you are being straightforward with your controls, you can have reproducibility in your experiments and you can publish. So I think I'll go to the figure to show this result. So here we have the graphs of our biolayer interferometry binding analysis. And up here we have our SARS-CoV-2, so the new coronavirus spike. And down here we have SARS-CoV-1, which is the original SARS coronavirus. And remember, for the original SARS coronavirus, the stronger the spike bound the ACE2 receptor, the more pathogenic that spike was, or that particular coronavirus was. So here you can see these square brackets usually means the concentration of whatever chemical is inside of those square brackets. So here you can see the concentrations of the human ACE2 receptor that they put into the solution. And so here you have a high 33.3 nanomolar solution of the human ACE2 receptor. And with that, you could see that a large portion of your, excuse me, of your, what's it called? Your spike protein ended up getting an ACE2 attached to it. And so you can see the higher the concentration, so here is 33.3 nanomolar, here is only 11.1 nanomolar, the higher concentration of human ACE2 ended up resulting in a higher signal than the lower concentration. And so by doing these different experiments, you can estimate the binding affinity of these two molecules. So here's the original coronavirus, and here's the new coronavirus, SARS-CoV-2, and here's the original coronavirus, SARS-CoV-1 from 20 years ago. So here you can see at the peak, after 1,000 seconds, you ended up getting a signal between 0.9 and 1 for the new coronavirus at this 33.3 nanomolar concentration. If you go to the same 33.3 nanomolar concentration of the original coronavirus, you can see that the signal at 1,000 seconds is only 0.3. So, or 0.33, we'll say. So all other variables controlled for. We can argue that this new coronavirus binds much better than the old coronavirus. And that's what you're getting here. And on top of this, I also want to mention, and also it's happening for all the concentrations. So at 11.1, you're getting about, 0.6 for the new coronavirus, and 11.1, you're getting about 0.15. So this is reproducible with the different concentrations of the human ACE2 receptor. Now, this is the part that I'm actually a little bit more concerned about, is that look at the receptor after it's bound and you do the wash. And you do the wash for the original SARS coronavirus, or the new SARS coronavirus spike and the old SARS coronavirus spike. As you do the wash, look at how quickly the ACE2 receptors are falling off of your spike protein for the old SARS coronavirus. And that's happening here as well. It's happening here. Now look at the new SARS coronavirus. You do the wash, and it's an excruciatingly slow process to get back to equilibrium. So what this can tell you is that after the binding domain of the new coronavirus, after that binding domain interacts with the ACE2 receptor, it has a very hard time coming off. So even if you were to completely wash away all the other ACE2 receptors away, they are going to stick very hard, and they are not coming off. So what this reads to me is that after, and I should also say, in comparison to this other binding domain, where you wash it and then it just drops off. What this tells me is that after the binding domain of the new coronavirus, after that interacts with the ACE2 receptor, what probably happens is that there is going to be some change in the shape of the spike protein or the receptor. Something changes, something moves around, and then it makes it even harder for it to release after the fact. This is just a intuition. This is my intuition speaking right now. So obviously this isn't necessarily, proven at all, but my intuition would indicate to me that sticking, yes, that is the random process of things moving around and they stick together and then they stay stuck together. But the process of washing and removing is so much slower in the new coronavirus that indicates to me that there is some other mechanism helping move stuff around to maintain its position that way. And the problem with this, the problem with this is that means that this new spike protein that is in the current virus causing the current pandemic, it sticks so incredibly well to the ACE2 receptor to the point where it might actually be greatly influencing the structure of the ACE2 receptor itself. So, and this might not necessarily have been the case with the old coronavirus. And that could have, implications not just with entry into the cell. That could also affect cellular signaling in some way, shape, or form as well. So just keep that in mind, that this new, they say that they have comparable affinities to each other, but these graphs don't tell me that they're comparable. They're comparable in that you can compare them to each other, and they fit on the same graph, but this does not speak comparable to me, at least. Oh, I need a drink after that. So, and I don't know if I mentioned that, but I'm actually going to stop at the discussion, and I'm going to do another video about that. So, going back to here, this is where we're at. So, and okay, the affinity determined here for SARS coronavirus spike soup binding to the human ACE2 is in line with prior studies, and the tighter apparent interactions formed by SARS-CoV, the original SARS-CoV binding domain, compared with the subunits, so the whole units, yeah, so it's in line. It's they're saying that they are reproducible. They're saying that they are able to, that their results for the original SARS are in line with other studies. Therefore, their results for this new coronavirus spike binding domain should also be taken seriously as well. So previous structural work identified 14 positions that are key for binding of the SARS coronavirus S-soup binding to the human ACE2 receptor. And these are the amino acids. Okay, so just so you know, the letter denotes a specific type of amino acid, and then the number associated with it is the numbering position that amino acid is in the protein. So for here we have threonine 402. So what they're saying is that in the position number 402 of the original SARS coronavirus spike protein, it was the threonine at this particular site plays an important role in interacting with the human ACE2 receptor. And then the same thing goes here. The arginine at the 426 site of the SARS coronavirus spike protein interacts well with the human ACE2 receptor. And anyways, analysis of the 144 SARS-CoV-2 genome sequences available from the Global Initiative on Sharing All Influenza Data, or GSAID, shows that 8 out of these 14 positions are strictly conserved in the SARS coronavirus 2 binding domain, whereas the other 6 positions are semi-conservatively substituted. So, so what they're saying is there were 14 positions in the original SARS that were highly conserved and interacted with the ACE receptor strongly. They could find specifically what amino acid on the binding domain caused that interaction. So, and that was conserved in the original SARS. When it comes to this new SARS, they say that eight out of these 14 positions of these specific amino acids in the binding domains, those were conserved. Those were the exact same amino acids. However, but there were 6 of those that were changed. They were semi-conserved. So that probably means that there was a little bit of, even though an amino acid changes, it might change into a similar amino acid that isn't exactly the same. So that might be the reason why they said semi-conservation of these other amino acids. And there's a figure that shows these that I'll show later. The conservation of many key contact residues could explain the similar binding affinities of the SARS coronavirus 2 S sup B and the SARS coronavirus, the original S sup B for the human ACE2 receptor. No mutations of residues predicted to contact the human ACE2 have been observed among the SARS coronavirus 2 spike sequences available to date. So what they're saying is, you have your ACE receptor, you have your spike of the new virus. Excuse me. All of the predicted amino acids at this interface are conserved as of the writing of this particular paper. They are all conserved. So that indicates that whatever interactions have been made already seem to be an optimal configuration as of yet. So you can have some mutations that are happening elsewhere. That's possible. But as far as the amino acids that are in contact with the ACE2 receptor right now, seem to be very well conserved. All of them. So all of the amino acids that touch each other are conserved. However, we note that SARS-related coronavirus, ZXC21 and ZC45, which harbors the most closely related spike sequences after this SARS-related CoV from this other bat, they have deletions in the receptor binding domain that could affect binding to the human ACE2 receptor. And this is between the SARS-CoV-2 S residues between 473 and 486. Collectively, these results suggest that SARS-CoV-2 is at least well adapted to the human ACE2 ortholog as the 2002-2003 epidemic strains of SARS coronavirus, which could explain the efficient transduction efficiency mediated by the respective S glycoproteins. And these are the figures, and the current rapid SARS coronavirus 2 transmission in humans. So, let's go to the figure. Oh, that's not the figure I want. I wanted this figure. Here we go. So this is the amino acid sequence of the receptor binding domain of the new SARS coronavirus and the old SARS coronavirus. And here, where you see an asterisk or a star, these are the 14 sites that were absolutely conserved in the original SARS coronavirus. And as you can see, it was 6 and 8. So I think it's so 1, 2, 3, 4, 5, 6, 7. So 8 of them are conserved in the new coronavirus. And 6 of them are completely different or semi-conserved in the new coronavirus. So wherever it's labeled red, that means it's the same, and wherever it's labeled white, those are that is a difference between the two binding domains. And remember, the binding domain of the new SARS coronavirus is significantly better at binding to the ACE2 receptor than the old ACE2 or the old SARS coronavirus spike protein. So I want you to notice that in a lot of, so in the sequence in between here and here, where the sequence in between these really important, or I should say the areas where you have a lot of these highly conserved sequences in the original SARS coronavirus, this area has a lot of mutations in it. And it's more mutation prone than outside of this area. So here outside the area, a lot of red. Here outside the area, a lot of red. And here within that area, you have a lot of white, or a lot more white than the other areas. So this is harkening back to earlier in this paper. It said that between the spike protein of the original SARS coronavirus and the new SARS coronavirus, that they are about 75% identical in amino acid sequence. But in the receptor binding domain, they are only about 50%. So there was a lot more variation, or a lot more of a difference between the two. So, and here, I just want to bring attention to the fact that 8 of the well-known amino acids that were vital for the original SARS coronavirus, those are conserved in the new coronavirus. So those are presumably still working to help bind with the ACE receptor. But then a lot of the, I'll say, the less important amino acids have been changed around those. So what does this tell us? This tells us that a lot of the amino acids that were considered non-important in the original SARS coronavirus have changed. They are now different. And so those differences could mean making it worse or making it better. Now, since we know that the current binding domain of the new SARS coronavirus 2 spike protein, since we know that that does bind a lot better, this could tell us, this could indicate to us that out of all of these new, I don't want to call them mutations, but all of these differences from the original spike, if you add up all of their effects together, you have a much better receptor. You have a much, or not better receptor, but you have a much better binding domain for that receptor. So I just wanted to highlight that a lot of these mutations, or not mutations, but a lot of these differences are happening in between what is considered the important residues of the original SARS coronavirus. So just keep that in mind. So we're going down. I think I should stop soon. Yeah, I think this will be a perfect place to stop. So the next location, the next part of this paper, and man, it takes a long time getting through papers, but the next part of this paper is going to talk about the architecture of the SARS coronavirus 2 trimer. And so what they did here, I'll just do a quick preview. What they did here was they just took the outside domain of the new SARS coronavirus spike, and they did a few mutations. So they got rid of the furin cleavage site, because with the furin cleavage site, you basically remove a big portion of these spikes. So they wanted to leave that in. And then they also put in a trimerization peptide. So they put in a part on the protein that is guaranteed to form trimers and guaranteed to stick together. So by doing this, they were able to make a lot of these structures. And then what they did was cryo-EM, which is you have a particle, a small particle with a bunch of these proteins in it. And then you freeze it. You make it very cold so that nothing's moving. And then you do electron microscopy on it. Now, electron microscopy is not not able to see these individual structures. But what they do is they take thousands of photos. If you have one drop and you have thousands or more, maybe I don't know the exact numbers, maybe between thousands and millions of these structures, what they'll do is each one of those structures will appear as a little fuzzy blob in the electron microscope image. And now we have developed algorithms that can look at thousands of these blobs, and then it deduces what structure can cause all of these blobs. So that is an elevator pitch of what cryo-EM does, or as far as cryo-EM being used to image structures as small as proteins. So I think this is going to be a good spot to end this video, because obviously 2 hours of talking is not good for me, my voice. So yeah, if you have any questions, go ahead, leave them in the comments, and all 3 subscribers of you. And yeah, I will continue on with this architecture, the architecture and the discussion of this paper. And I will also leave a link in the description to this paper if you want to read it for yourself. But yeah, there we go. So yeah, thanks for coming.